sample is here, the sample actually gets washed, filtered, and goes into the lab. And then the reagent acts, the reagent will come from here, will wash the DNA, go into dump. Finally, the elution buffer will come, take the nucleic acids and elute it out over here. Then the cartridge comes out,
Comes out.
and then you
Okay.
collect.
So once the extraction is done, it releases the cartridge. Good. So next stage,
Next stage, you collect the nucleic acids,
okay,
100 microliter.
collect it from this cartridge.
So along with that, she will give the pipette.
So where's the pipette? Can you show me? I saw the pipette. Now, yes,
not
not
this
this one. Okay.
So it will be a separate
A separate pipette.
pouch.
Okay, good.
Pouch. So you collect it into the end of tube, which will be also provided along with
Also
the tube.
provided. Okay.
You collect it, and that thing you can also store it as much as long you want it. Around -20 or to 20 degrees Celsius.
-20 to 20 degrees Celsius. So it has to be refrigerated. It will be frozen or it will be cool.
You can also use it immediately if you want to test it.
So -20 degrees Celsius to 20 degrees Celsius. So the laboratory must be equipped with a refrigerator. If they are not using it, if
It's
it
not
is
a must. It is only when you want to store it.
Your sample.
sample, okay,
Otherwise, you can immediately run it.
you can run it immediately.
So once you extract it, this will take around 20 minutes.
Good. Now, is it powered by batteries?
Yes.
So no power failure. Even there's no electricity supply,
It continues.
you can run it. Brilliant.
Yeah.
And this is what we need.
Yes.
Yes. So then we move to
Then you have a disease specific chip. What particular disease you want to test it for?
okay.
Like if you extract serum sample, there is a possibility you want to do a HIV test, HBV test, HCV
So
test.
for every single disease, there is a separate kit, chips for them, right?
So these chips
Good.
will be like this. So these chips will go inside this device.
So I can run 4 tests at a time on the what did you call this? Quatro. On the Quatro, 4 tests at a time. So let's say I want to run human papilloma virus, hepatitis C, HIV, malaria, or something. We run the 4
Independently.
So you apply the sample.
You take
In
with the
this
help
well.
of 6 microliter
Let me
pipette.
see. Can you see? No, show it here. This.
You have to load it on this
Okay.
6 microliter. This goes
Six,
inside.
6 microliter.
So this will go exactly
So let's see how it works.
here.
Open, so it opens the tray. Can you see? So you select the test you want to
Select the
do.
test
Bring
you
it
want.
closer.
Select
You
sample.
select
You
the
select
test you want
sample.
to
You
do.
also
You
select
want to
test.
do MTB, you select MTB.
So
Like that.
okay, so you want to do chlamydia and gonorrhea or something, you select, right?
It will
And then insert the chip,
Yes.
then it starts running.
Once
So
you start running, then automatically, you'll get at the end of 45 minutes. you'll
how
get
many
the
minutes?
45.
45 minutes.
Yes.
Is a turnaround time.
Yes.
Wow.
So within 45 minutes, you are ready with the results.
Okay.
So in result section, you will get the control, which will amplify anyway.
Okay.
And if the blue line, that is the sample
Bring it
target
closer.
gene.
Okay.
If it
So
also
they
amplifies, it will give positive result. If doesn't amplify, goes flat, it will give you a negative or not detected.
so
result.
let's come again. The control is the amplified.
Control comes from here.
Okay. Yes.
So
Okay.
your process control goes along with the sample, and it is validated over here.
Okay.
So process control will also amplify along with the sample if the sample is positive. So the red line is the control,
Red line is the
the blue
control.
line is the target gene.
Blue is the target gene. Okay.
So in this case, the red line is amplified, that is the control. So
Okay.
it validates the process.
The process,
of the
yes, yes.
extraction.
Once the control is there, we know the process
Once
is
that is
good.
done, then you have to see whether the blue line, the target
It's
gene is
also
amplified
amplified or not.
If it is not
Okay.
amplified, that is not
That is
detected.
negative.
If it is amplified, it is detected.
Now, is the result qualitative or quantitative or
It will
both?
depend on what kind of a sample or what kind of a disease you are doing. For example, if you are
Hepatitis B.
doing the influenza AB or COVID-19, your results will be qualitative.
qualitative for
If you are-
COVID-19
yes.
or hepatitis B,
Hepatitis
HIV,
B viral load, it will be always quantitative.
quantitative.
It will depend whether it is an IFU or whether it IU or whether it is into CFU or whether it is into copies per ml. You can also convert from IU to copies per ml in this device.
So now, what is interesting here is that people in the villages can enjoy quality tests with your product, right?
We are already doing that.
They're already enjoying it. So now we have this briefcase, if I'm correct. So it can carry everything. So this is the extracting chamber, right? And then this is the for single channel testing, and then the printer is here. Fantastic. So this is quite portable. So you can have it all, in this container and take it anywhere. So people are already enjoying access to healthcare, even where there is no laboratory. You can get quality testing.
So presently we have 40 installations into Stop TB program, which is into Nigeria. So these are the states.
These are the various places where
It has
this
been installed, we've been deployed.
great product has been deployed. I can see my state here, or your state.
That's it is there, yes.
Even Lagos State.
Okay.
Ibadan
Yeah.
North East local government area. So these products are there. Taraba, right? Akwa Ibom.
Yeah.
Wow.
Katsina, Bauchi, Kano, and all
And
that.
it
They
is
are all
not
there.
only for Nigeria, it is for
It's
Africa.
also for the
So,
whole of all these other
Across
countries
Africa
and across the world as well.
and across the world. So testing is becoming advanced and accessible.
Accessible, and it will have a wireless data transfer.
Okay, so in terms of data governance, so there's wireless data transfer. So it stores memory, I believe.
Yes.
Good.
50,000 test results can be
50,000 test results.
and this can be deployed into doctor's office, pathology laboratory, mobile clinics, primary healthcare
And
centers.
of course, that is what we are saying on our training program, point of care testing.
Yes.
We are letting people know that the limitations are being removed by technology.
And this
Right?
will-
Doctor's office, so no need to start looking for a lab or stressing the patient. A lot of people die because results do not come out on time, and doctors cannot make the right decisions fast. So doctor's office, pathology lab, right? Mobile clinic, yes, you can take advantage. Research institute, primary healthcare center,
And more important is this device is endorsed by WHO
endorsed by
for
WHO.
TB and MTB ref.
Good, good,
Drug
good,
resistance.
good. So I'm happy that we're able to provide care for people, bring it much closer. Because before now, people believe that you can only get equipment like this in national hospitals or in teaching hospital. But as we equip our primary care centers
Yes.
with advanced technology like this, then it makes diagnostic more
Yes,
accurate
and accessible.
and accessible.
And it only doesn't do the TB diagnosis. It can do around almost 35 different parameters.
Yeah, 35 different parameters.
With others under development. Those products coming soon, actually products that are already undergoing clinical trials from one stage to the other. Some of them are already done validation. Some of them are getting certification. So once this is done, the products are rolled out. So coming soon is actually coming soon. It's not the coming soon that you hear from them.
Okay.
So these ones are the ones in the pipeline. We have
These are
some
the
products
ones.
in the
Can you
pipeline.
move back this a bit?
There are those we have in pipeline. Those
So
are the
these
ones we
are
are
available now,
Yes, currently available.
about 30,
35. Yes.
35.
Yes.
And I can see a lot of diseases of public health importance.
Yeah.
Cholera,
Yeah.
COVID-19,
Yeah.
HIV,
Yes. Malaria.
malaria, and all of that. Because there are times people do malaria test, they say it's negative
Yeah.
or something they're not sure. They can rely on them. So these are in the pipeline.
Yes, they are not in the
waiting for-
pipeline. They are already coming soon.
They are coming soon, okay. They
Clinical
have been
trials
approved
done,
and all
approved,
that.
and all
See,
that.
from the Nigeria's point of view, some of the diseases like malaria, is quite common over here. So malaria chips are available with the differentiation of vivax and falciparum.
And falciparum, okay.
So you can differentiate
so
between
know
that.
which disease
Yes.
is responsible.
Yes.
And I think we're having a few cases where people test positive on RDT, the rapid
Yes.
test
Yes.
for the pan antigen.
Yeah.
But you are not sure if
Which
it is
of them,
which of them.
Yeah.
So this
Yeah.
can tell us if it's vivax or falciparum.
Yeah, and
That's
recently
different.
Nigeria also had a lot of new diseases coming like chikungunya. So when you do the chikungunya test over here, you can see the last one. So you can differentiate between dengue and chikungunya on a single chip.
So not all fevers, not every fever is malaria.
Yeah.
It could be chikungunya, it could be dengue. Yeah,
So
that's
these ones that you can see, including Ebola,
Ebola.
those are the ones you
Okay.
can see that we are
Pipeline.
develop probes and primers for them.
Okay.
Then when after some time they move to the coming soon, when you are sure you have done clinical trials and just waiting for certification.
And as I said, it's WHO endorsed. 5 minutes for the sample collection, 20 minutes for the extraction, 35 minutes for the analysis, and done.
There you are done.
Yeah,